Hello, in this video we're going to be talking about transcription. So let's not waste any of your time. Let's get right to it. Okay! Now when we're talking transcription, the first thing we want to recognize is that we're talking about taking a DNA strand and reading those individual nucleotides and matching up their complementary base pairs to form an RNA strand. So if it's an A, then the RNA strand will receive a U. If it's a C, the RNA strand will receive a G. And if it reads a T on the DNA strand, then it'll put an A on the RNA strand, and then vice versa with the C and G. If it reads a G, it will put a C on that spot. Now this is done by a lot of different things help with this process, and also based on what type of RNA you're making determines what different polymerase is going to be working on. If it's just your ribosomal RNA, I lost my word there for a second, then polymerase 1 is the polymerase that does that job. If you're making mRNA, which is what we're talking about when we say transcribing RNA from DNA, then polymerase, I misread my line. That's polymerase 1 does that. No, I'm sorry, 2. I'm sorry, I misread my line the second time. And then if you're just making the tRNA, which you may not be familiar with right now, but you will be by the time you're done with this series, the tRNA is the individual component that brings the amino acids to the RNA strand. They're like little amino acid carriers, but it is a strand of RNA, a type of RNA. And when you're making those, and polymerase 3 is one that makes those. And each polymerase has a different promoter, a different way of starting, but we'll touch on all that before we end this series. Now, the idea of transcription, this is a 3-step process. Initiation, which can be synonymous with beginning, the starting of the process. Elongation, which is simply the building of the RNA strand as it goes nucleotide by nucleotide, reading and matching those complementary base pairs. And then termination is when it gets to the end, it reaches a particular DNA section, and that's coded in a particular way. We saw the stop codons on the last video. It runs into one of those, and that means, okay, we're done here. Let's stop, and everything drops off the RNA strand, and then the next process begins. But 3 steps process, and obviously the first one and the last one are just start and finish. So if you can imagine, if you were running a mile, you'd have your start of your mile, and you'd run the distance, and you'd have the end of your mile. So the start and the ending are pretty quick, short processes. It's the middle one, the actual running of the mile, that would be the length. So it's a majority, almost 99% of the amount of time is during the elongation. Now, when I mentioned there was a lot of components that go into part of this, the initiation process is done after several things do all their pre-work and get things prepared and get things set up. But you've got your transcription factors, which are part of the initiation complex. And by complex, we mean just set up all the workers attached to the RNA strand kind of in one general area to get things started. And each transcription factor will have a different kind of job or chore or task, but it's all designed to get everything ready for the process. And we looked at the last video lecture series when we talked about, I just lost my thought there, so I'm going to move on. But the idea of like making the replication fork and getting everything organized for replication, that's what it was. A lot of things were involved, the same thing as here. There's a lot of different enzymes, a lot of different processes. Some open up, you've got to open up the DNA strand, you've got to close it, you've got to read, you got to proofread, all that stuff. So there's just a lot of things involved in what's going on. But it's not so important to know that which factor does what, or else I would have listed, or since I didn't list it. But it's more about the transcription factors are part of the initiation complex. Some other things to recognize about that RNA strand. Once it's made, it's got to go through some modifications to make it more similar to the DNA strand. If you'll think back to DNA, when we talk about replication, we talked about being read from the 5' end to the 3' end. Well, those prime ends, those 5 and 3, they're particular sets of molecules, a particular molecule. And so once the RNA strand is complete, then you've got to add that 5' end. Excuse me. And the 3' end and the 5' end, as you can see there, is a sugar with some phosphates and a methyl group. And the 3' end is just a series of A's. And the stop codon is typically right in front of that. So those things aren't read. They don't express anything. They're just part of the structure of an RNA strand. And those same things are what's on the ends of the DNA strands. Now, some other modifications that occur. There are sections of the RNA or the DNA strand that are not read. Okay, those are called introns. They have their own functions on a DNA strand as far as the well-being and structure of the chromosome, but they're not read. They don't code for anything. So once you get to the RNA strand, then those are removed. You can see those are purple things inside in between the yellow, and those are simply those are simply removed. Splicing, as you can see, there are some other things that are removed, but certainly those are. And you've got a particular enzyme that is there. That is its job. Okay, one more thing that we want to think about as far as this scientific research and understanding genes and their expression. Well, part of that also is understanding what everything makes, what they're a part of. And we want to think about RNA strands that are made. There's lots of varieties, as we already talked about. But polymerase 1, you can see what it makes there, and then polymerase 2 and 3, and as it says there, be sure to record that in your notes. But also, if you want to think about application-based, and this is kind of a peek at, this obviously is a eukaryotic idea, but you can think about it in a prokaryotic idea. If you have particular polymerases, if they're a part of the building the RNA strand, which then leads to making the proteins, if there are things that kind of make them not work as efficient, maybe they're sensitive to, in this particular, the amytin is a product of a fungus, and it simply goes in, and if the polymerase is sensitive, in other words, it affects how well it works, then you can have a breakdown in the protein production. If you think about that application based to the prokaryotic cells, if you can find a product or a molecule of some kind that slows down or stops a polymerase from working and doing its job, then you can find a way to use that against a prokaryotic cell. You just have to make sure that it doesn't affect us too. You wouldn't want to take an antibiotic cell that breaks down nuclear membranes, because it would break down our nuclear membranes as well. Well, that's our video over transcription. When we come back next time, we're going to be talking about tRNA. We'll see you then.